So today we are pleased to welcome Dr. Glendon Parker from the University of California, Davis. Glendon is an adjunct associate professor in the Department of Environmental Toxicology at UC Davis and the inventor of protein-based human identification. His research career began at Monash University in Australia, where he obtained his Bachelor of Science degree in biochemistry and thereafter his PhD in biochemistry and molecular biology. In the past, he has worked closely with the Forensic Science Center at Lawrence Livermore National Laboratory, and he has a history as an academic and life scientist, including at Utah Valley University and University of Utah. He is actively engaged in his research and development of techniques that obtain useful genetic data from forensically informative proteomic datasets. His major focus currently is on developing products to use the hair shaft of proteome to obtain genetic measures of human identification and biogeographic background, but also includes bone tissue and sexual assault kits. As a well-developed lab, Parker's group at UC Davis is working to establish proteomics as a new tool in forensics and anthropology. I will now pass my screen sharing to you, Glenn, and you can begin when you're ready.
Thank you, Casper. Let's find our, here we go, where is it? There we go. Are we up? So I'll just go to the top here. So does everyone have the screen up? Let's start this now. Okay. So I'd like to start by thanking the organizers at PEAKS for this opportunity to share our work. We don't want to jump things too much. And the opportunity to speak and share our work with you today. Just as a disclaimer, this is my personal opinion and not those of any of my sponsors. And I do have a conflict of interest. This work does have some intellectual property associated with it. So I'm not an anthropologist. I'm a biochemist with a history in background in the life sciences. So I'm coming at this from a scientific point of view. And from a sort of a scientific point of view, in terms of analyzing, say if you were to put yourself in the place of an archaeologist or physical anthropologist who's actually digging a site and comes across some human remains, there's some things that we can actually get some good information about. Looking at their anatomy, we could basically get a pretty good idea about how old they were when they died. We can look at the context around them in terms of any grave goods they have. Using isotopes, which my colleague who's here today, Yelma Erkins, does, we can sort of look at where they came from and get an idea about migration and things like that. A careful look at the anatomy and also at sort of biological molecules. We can get an idea of any disease status they had. Of course, we can do carbon dating. We can obviously do sex, species identification. On an older material, there's a lot of people who are working on phylogenetic questions that you can get from these biomolecules. And of course, you can estimate sex. And sex is, well, I guess all of these are things which are accessible to a scientist, but they also give you a framework with which you can ask bigger questions. And these are anthropological questions which don't necessarily have a distinct readout, but these are, I would think, these bigger questions are what motivate us to get in there and to actually examine these materials, to treat them with respect, and to try to get an idea of the sort of the cultural environment that these individuals came from. What were their lives like? How did their culture treat them? And how did they view their own culture? So the knowledge of the sex of that individual is a major part of addressing some of these questions. And I'm just trying to move forward now. Here we go. So up to this point, there's been sort of two main ways of looking at this and addressing this. The most common and by far the most historical is the use of the actual skeleton itself. There are anatomical features that are distinctly male or female, and I'm pointing there, you can see in the top left the pelvic arches there. The pelvic anatomy is very distinctive. There's other areas of the body, such as the degree of robustness about the skeleton, which could also be an indicator of the sex of the skeleton. The other more recent sort of additions where people are looking at DNA, both sort of just reading DNA straight like we have here with the standard STR CODIS kit. This is more recent since the sort of DNA revolution, and of course there are a lot more recent advances with ancient DNA that are more sophisticated here. The other element that we have that we would like to introduce today is a third fundamentally different way of looking at sex of a skeleton, and that is to use proteomics. And specifically, we're looking at the Amelogenin gene family. So what's the advantage for doing this? Why would this be necessary? Unfortunately, the anatomy of an individual up to the age of puberty is very, very similar to the degree that you really cannot have any degree of certainty about what the skeleton is in terms of sex. Also, a lot of skeletons are degraded. The pelvis is one of the softer bones and is more likely to be degraded. So in aggregate, about half of the skeletons we have are less accessible to sex estimation from that. Likewise, DNA is less stable than protein. DNA, as we can sort of see here in the bottom right, has a lot of oxygens and nitrogens in its molecule. And so environmental chemistry will come in and react with oxygens and nitrogens. And so each of these will degrade the length of the template to the point where it's very difficult to do PCR. And so with older samples, you're going to get some degradation. So the Amelogenin gene family, which was, I guess, used is one of the main ways, at least in forensics, of looking at whether an individual is male or female. It has basically a copy of a gene on the X and Y chromosome. And I have the two gene products here. In the rectangles here, we basically indicate the exons that are swapped in and out for various splice variants. And you can see here in the blue squares, these are amino acids that are specific for the Y chromosome form of the protein or of the gene. And so there's actually quite a few of them. And so this basically allows us to, if we could detect any of these amino acid differences, then that allows us to infer whether there was a Y chromosome present in that individual or an X chromosome. Now, what I think is a wonderful coincidence is that the most characterized sex-specific gene family up to this point is expressed in the most robust human tissue. And so that's, to me, a great coincidence, if you want to call it that, and allows us to now use this tool to address questions going back in time. So we have here an example. This is from one of our earliest data sets where we took some teeth, we processed them, we demineralized them. We then went ahead and did some, put them through a mass spectrometry workflow. There was a shotgun proteomic workflow. And this was the data we got. I think this is even from our very first set of experiments, our preliminary data. and we put them into peaks. Those of you will be very familiar with this sort of format. And I've indicated in these blue squares the amino acids that are unique to the Y chromosome form of the protein. And you can see that there's quite a few of these that actually are represented in these different peptides. The other thing is that there is a region where there's not a lot of difference between the two, and it actually is very well represented in the end, towards the N-terminal part of the protein, and that's sort of not unique. It doesn't sort of tell us, it's not informative as to which version it is. But past that point, and we sort of think of this isoleucine here at position 46, beyond that point, we start to have a high density of differences occurring. The other thing that I find interesting is that there's a lot of variation in different peptides here, and this is because of the biogenesis of enamel. As part of that process, there's a lot of endogenous proteases, and these tend to break down the molecule in a very sort of non-specific way. And so that any one amino acid, and you sort of think here of this sort of distinctive methionine here, is represented by something like 15 different peptides, and in some data sets there can be 20 or 30. And so it's a very diverse population, which is actually quite helpful in a way, because you also notice that a large chunk of the, chunk being a highly scientific term, a large chunk of the protein actually has not very many tryptic sites. And so this actually was fortuitous, and that allowed us to get information. So you're very familiar with this environment. I'm just putting it on to say this is sort of how we go about in actually quantifying this and how we process the data. So we put into peaks, we run it through. We did do, we of course naturally put all of the isoforms from these genes into our reference database. We go and do our search. We're pretty stringent. We stick to sort of standard methods here. We then go through, identify the relative gene here, Amelogenin Y. You can see here that we have specific peptides. We then get the peptide list and organize it in terms of we basically arrange them in terms of the sequence that they appear in the protein. And basically everything that is, that includes this isoleucine 46 and below is pretty specific. There are a few examples you have to keep an eye out of smaller peptides that are not specific, but pretty much there's a demarcation here. So we basically take this, we add up all of the intensities and pull them, and that gives us a measure of a label-free quantitation. We do the same for the X chromosome, go through, do the same process, and then we can basically, we have two data points, and we see that there's two different populations that form. There are those that have both the Y chromosome and the X chromosome, which are males, and then we also have females which don't have any of the Y chromosome isoform, but have the X chromosome form. So you can see a few things here. One is that there is a range in the signal. There's about 2.5, 3 orders of magnitude here for the Y chromosome, and about 4 orders of magnitude for the X chromosome form. We think this is biological. We see this in different sort of ages. So there's obviously some interesting biology happening here as to why some individuals have more of this than the other. The other thing to note is that we include in this data previously unsexed deciduous teeth that we were able to then assign as male or female. This is something which can't be very easily done. These were actually teeth that the male teeth were actually isolated teeth that were found in a field in southern Illinois. So there's zero context behind these skeletons. In fact, teeth were the only thing that we had. And the other thing is that there's some clear misassignments too. We see here that this is something that had previously, you can see that there's two blue squares here in the female grouping here. And so one of these very clearly it was an incorrect misassignment, and then another one was a sort of a low signal individual or data point. And so the question is, what's going on there? Is that a false negative? And so that's sort of an additional question that we want to address later. The point that we see misassignment sort of leads us to realize that now we have three methods of sexing a skeleton. We have the potential of having sort of conflicting data sets. So let me just continue on. So we have the signal is very stable over time. So here on the left, you can sort of see, this is exponential, and this is now a few years old. But you can see that range that we have for the signal is actually pretty stable. Even very old samples have that same range. And so we found this to be very interesting. This is a biological readout, and it is just as stable, and we're getting high quality data on samples that here are 7,000 years old, and we've since done 9,000-year-old samples as well. So the stability, I think, is remarkable. And granted, there's probably a few of these. At some point, we're going to get degradation, but this is just a remarkably stable signal. And part of the explanation, we think, is here on the right, that this is the percent of the total proteome that is in these amelogenin peptides. And we can see that over time, it is increasing. And so some of these older samples are up to 70%. So I would put that the stability of the signal is probably due to two factors. Amelogenin will be degrading, but it's degrading at a slower rate. And I think it's because it's associating with the minerals, the two-dimensional interfaces that are going to be present there with the calcium apatite. And this sort of builds on some really, I think, some groundbreaking work by the Collins group with ostriches and things like that. And more recently, very similar work to this, but with much older samples by Enrico Capinelli and Frido Welker. So we took these three different methods, and we actually had an opportunity here with some colleagues of us with Far Western Anthropology group in Davis to investigate two contexts that were very close to each other here in the East Bay in California. Total of about close to 100 or so, 105 burials. We took 55 of those and processed them. All of these burials were processed for osteology in terms of sex estimation, and also they had ancient DNA conducted on them as well. So this gave us a really good opportunity now to do all three of these methods on a large size cohort. And so we were then able to directly compare the sex estimation that occurred in these samples. So the advantage here is when you have a large size of cohort, 55, and you have three methods, you can now start to develop a framework to resolve differences that could occur between them, if there's any inconsistencies in the data between the sex estimates. And if so, why? What is going on? What are the conditions where you start to see inconsistency starting to develop between the two different types of data? So we went through and we mapped them all out, and we ended up having something like about, I think it was 12 or so different, depending on the various methods we used, different results from the DNA and from the proteomics. So we have here plotted out so there's two ways of sort of doing DNA sex estimation using ancient DNA. There's a method developed by Pontus Skoglund, who basically looked at the ratio of the X and Y chromosome reads, or Y and X chromosome reads. And then there's also a more recent, and in my view, slightly more robust method with Elisa Mitnick, who was basically looking at the X chromosome to all the other chromosomes. So when you plot them out, and this is sort of how Pontus Skoglund actually went through and sort of framed his data as well, you can see that, and then we can start to look at these conflicts. There were basically three scenarios that formed. There was basically, we have full agreement between the proteomics and the ancient DNA methods. We have where one of them gave us no assignment, where the DNA gave us no assignment. We did get proteomic estimates, and actually in this context, in this particular experiment, all of the proteomic estimates were 100% consistent and gave us good data. But what's of interest is that there are some that were conflicting. And so we see some here. There's these are ones which are sort of clustering in the female cohort or population, but these were distinctly male samples here on the bottom left in gray here, based off the proteomics. And then there's a few where we see inconsistencies in other directions. They're sort of clustering with the male samples, but they give us, these are very clearly female individuals based off the proteomics. And so if we plot them out, these consistencies and inconsistencies, we can start to look at patterns. And the pattern that we see is that below 100,000 reads for the ancient DNA, this is where the errors start to take place, or inconsistencies. I don't want to bias things by saying errors at this point. Above this point, it's very robust. And I can pull up the Mitnick data later if you want to have a look. But the Mitnick data was a little more consistent when it had definitive calls. We basically start to see that there's errors starting to take place here, and it's below 100,000. Now, a lot of these are conditional estimates. They're consistent with data, but not all of them. So if we take the same data points, but now plot it out on the proteomic data, so here we see what we saw before. In the bottom left, we see the Amelogenin Y readouts and the Amelogenin X readouts. They basically separate out into the male and the female population. And here we don't really see much of a pattern. The indeterminate ones are spread throughout. You could, why is it we have a cluster here? That's something we've thought about, but I would hesitate to say that there's a pattern of sort of inconsistencies when you sort of are looking at it from a proteomic point of view. So from that, I would say that because of that, I would basically state that the proteomic estimates are going to be more robust and reliable than the DNA estimates. And we can put that into the context that if it was, for instance, a contingent DNA estimate, consistent with result, if it was below 100,000 total reads, well, that's when you should be questioning your data. Above that point, it should be considered totally reliable. And we see that here when we sort of mapped out these differences. So if we're looking at just the inconsistent conditional estimates, we saw conflicts between proteomics and the ancient DNA at a very high rate when you're looking just at those consistent with results. And I would say it's only just slightly better than random. So I'd like to just refer you here in the bottom right. We can see that there's also that other point about a low signal sample associating with the female population. Is that really female, or is it a male false negative? So we spent a bit of time sort of trying to get a framework for this. There's several ways to approach this. We approach this as sort of setting up a logistic regression curve where we assume that it would be a symmetrical sigmoidal curve, and that as you get better and better Amelogenin X peptides and get more and more of them, the probability that it would be a false negative male result would go down, and that the probability of female is directly as a result will go up as you get more of the female signal. Now, we have looked back at this and we think that there are a few issues here. One of them is that our data is too good. We have very few samples that are actually very clearly male from a DNA, high quality DNA point of view, and actually is showing up as female in the proteomics. In fact, we've done this, I think, I would say we've done 250, 300 sort of teeth at this point, and we only have one sample that is very clearly falls into this category. And so we obviously need to sort of think about this and do some other modeling and some more development here. There's other ways to go about this in terms of quantitative proteomics. There's better ways perhaps to address this. But I think this sort of overall framework, if we were able to populate it with additional data points, this could also be a robust result as well, because male and female are pretty categorical classifications, and so logistic regression is probably the best way to address this. So in conclusion, in our hands, Amelogenin peptides are very stable. In this second experiment, we had 100%. In the earlier data, it was close to 100% readout. It performs better than ancient DNA, particularly if you're getting samples that are less than 100,000 total reads, which in this cohort was about 50% of the time, a little bit more, 60%. So in future work, we need to further refine the situation where we have a probability of female. Should female sex be expressed as a probability? Then there's other modes of detection and other models of detecting these peptides.
I'd like to acknowledge my collaborators.
Oops, sorry. Are we done? I think I must have copied over something there. I'd like to thank Yelma Erkins, Tammy, Rippon, Brian for helping us with this project, Julia for a lot of the earlier work, Yelma, and also my current collaborators, Jane Bickstra, Ann Stone from ASU, and Jason King. So thank you, and with that, I think we're good. I think we had a few technical issues there, but anyway, I think we're good. And if there's any questions, please feel free to ask.
Perfect. Thank you, Glendon. That was a great presentation.